So now let's take a look at our experimental species, C. elegans, and see how we can illustrate this concept. The researchers in this experiment exposed three population types to mutagens to induce a high mutation rate. So we're going to be generating these slightly deleterious alleles at a very high rate to be able to observe their accumulation. The three population types are, on the one hand, the populations with the mutants that are all hermaphrodites, so zero sexual reproduction in that population. The wild type that includes both hermaphrodites and males is one of the other population types, and then they created populations that are composed entirely of females that have the mutation that caused them to not produce sperm, so obligate sexually reproducing individuals. So in these three population types, we're inducing mutations and allowing these deleterious mutations to accumulate over 50 generations. So they then compared the average fitness of the three populations after 50 generations and compared them with the average fitness of the first generation in each of those three population types. And here's what they found. For reading this graph, on the y-axis, we have this comparison between the fitness after 50 generations and the average fitness of the first generation. So negative means that after 50 generations, the population was less fit. Zero means it's equally as fit as the first generation was. And above zero means that the population was more fit after 50 generations. On the x-axis, this outcrossing rate refers to the three different population types. So you see that there are 3 points on each of these graphs. One refers to the obligately sexual, so the outcrossing is 100%. All of the reproduction that's going on is sexual reproduction. So this is a population where the hermaphrodite females are unable to produce sperm. The zero outcrossing rate is the population composed of all hermaphrodites, so that mutation that kills off the males is fixed in this population. And then the one that's in between is the wild type population that includes both hermaphrodites and males. In the populations that were exposed to the mutagen as compared to the controls that just underwent normal natural rates of mutation, the ones that were obligately asexual, only hermaphrodites, had much, much lower average fitness after 50 generations than the control group that were obligately asexual. We see a little bit greater average fitness, but certainly no decrease in average fitness in this obligately outcrossing population where the females don't produce sperm. And the ones in between are less fit, but not as much so as the ones that were just hermaphrodites. So I just want to point out also this difference between the outcrossing rate in the wild type populations. So this outcrossing rate is related to the proportion of males in that population. And the proportion of males is going to depend on the difference in survival rate between the offspring of sexually reproducing versus asexually reproducing individuals. So in situations where sexually reproducing individuals are more likely to produce surviving offspring, you're going to get a greater proportion of males in that population. You're going to get a greater outcrossing rate, and that is the case when they're exposed to the mutagen. So this is another indication that sexual reproduction is beneficial in reducing that genetic load when exposed to mutagens. As compared to the control that has a lower outcrossing rate, it's less beneficial to reproduce sexually under these circumstances. We see that the fitness increases just under the normal mutation rates. If they're forced to sexually reproduce, it's definitely beneficial. This point here indicates that balance between the numerical benefits of producing asexually and being able to reproduce more offspring without having to invest in males that are not going to be part of that reproductive output, and the detrimental effects of the accumulation of these mutations. The results show that the population with no males suffers decreased fitness due to the accumulation of deleterious alleles, otherwise known as genetic load, and the mechanism for this is Muller's ratchet, the removal by genetic drift of the most fit zero mutation multilocus genotypes. Recombination in the highly outcrossing populations restores those zero mutation multilocus genotypes and avoids this decrease in fitness over time. So this is one of the big benefits of sexual reproduction. So our other model for the benefits of sexual reproduction is this idea of natural selection in a changing environment, being able to adapt quickly to changes in the environment. And this is related to something that you probably heard about in your ecology class called the Red Queen hypothesis. So this hypothesis is named after the character in Alice Through the Looking Glass, the Red Queen, who has to run as fast as she can to stay in the same place. So this is analogous to the idea that if you're in a changing environment, you have to keep changing with it to maintain your same level of fitness. This is exemplified by coevolution, an evolutionary arms race between predator and prey, between parasite and host, herbivore and plant. Basically, your environment is constantly changing, right? If you are the predator, your prey item is constantly going to be evolving ways to elude you, to avoid being eaten. You have to keep evolving better mechanisms for catching that prey. Once you evolve better mechanisms for catching that prey, your prey item has to evolve more mechanisms for eluding you. And so this is this constant evolution, this constant adaptation to be able to keep up with these other organisms that are an important part of your environment, an important selective force. So what you end up seeing is that multilocus genotypes that are beneficial in one generation may not be beneficial in the next. And then they may be beneficial again in a later generation. Recombination is your best bet for being able to do that quickly. If you're able to recombine and scramble up, reduce linkage disequilibrium, right, in the population, you can restore those lost multilocus genotypes when they become favorable again. Recombination also maintains more overall genetic variability, so both recombination and the segregation of alleles, shuffling the deck constantly to maintain the genetic variability, and that gives you that variation that you need, back to Darwin's four postulates, to be able to evolve by natural selection, adapt to changing environments. So let's go back to our model system to see if we can find some evidence for this working. So this time, we're going to use just wild-type worms. So just the populations that include a combination of males and hermaphrodites. So our experimenters treated these populations of wild-type worms with mutagens for 3 generations to induce a large amount of genetic variability. And then the worms were raised in an environment that required them to cross what they call a lawn of pathogenic bacteria to find food. So these pathogenic bacteria, if the worms ingest them, can cause the worms to die. And so this is introducing a big selective pressure into the situation. And they used 3 treatments. The first one is one where the bacteria were there, but they were killed. So this was the control. In the second treatment, new bacteria were introduced each generation. So in this case, the worms evolve, but the bacteria can't because new bacteria from a non-worm environment are constantly being brought in the environment of the worms. And a third treatment is that the bacteria in the environment were taken from the actual dead worms from the previous generation. So in this case, the worms and the bacteria co-evolve. So this is going to be one of those evolutionary arm races between a parasite and a host. And these are their results. So on the x-axis, we just have generation. So this is just time. And on the y-axis, we have the outcrossing rate. So remember, as I told you in the previous experiment, the proportion of males in the population is related to this outcrossing rate. It's going to be a reflection of how many males there are. And that's going to depend on how much better offspring survive if they are fertilized by a male rather than a hermaphrodite fertilizing their own eggs. So let's take a look at these results. In the control, there's not any change over time in the outcrossing rate. It basically stays the same. If we look at the situation where just the worms are evolving, what we see is a rapid increase in the outcrossing rate initially as the worms are adapting to these bacteria and evolving resistance to them. Once they've done that, though, then they go back to having more hermaphrodites and fewer males in the population because at that point, once they already have the multilocus genotype that's optimal for facing these bacteria, now there's a numerical advantage to being asexually reproducing according to Haldane's model, and now it's not so beneficial to have the males anymore. But in the group where the bacteria and the worms were co-evolving together, so we've got this evolutionary arms race situation, you have steadily increasing outcrossing rate, and it remains high through all of the generations in the experiments. We can see here that in the situation with a constantly changing environment in the form of pathogens that are also evolving to overcome your defenses, now you want to be able to constantly reassemble these multilocus genotypes to have the most favorable combination of alleles at any particular time. So in conclusion, what's so great about sex? Through segregation of alleles and recombination during meiosis, sexually reproducing populations can reduce genetic load by recreating the more fit zero mutation multilocus genotypes, and they can also quickly generate new combinations of alleles to adapt rapidly to a changing environment. And we saw this demonstrated, both of these two mechanisms demonstrated in the same species. So both of these things are happening all the time in species that are able to either reproduce sexually or asexually. And so in conclusion, there could be situations where asexual reproduction might be beneficial, and that would be in an environment that is fairly stable, so that once you have a multilocus genotype in linkage disequilibrium, so those beneficial alleles are not going to be separated from each other. If your environment isn't changing, you want to stick with that combination of alleles, right? So asexually reproducing is the way to go because you're going to exactly replicate that highly successful combination of alleles in your genotype. But as soon as that environment changes, if you're reproducing asexual, you don't have a good mechanism for generating a lot of variation very quickly to be able to deal with that changing environment. So sexual reproduction is going to be favored under situations where conditions are changing and you need to quickly adapt to those changing conditions.